Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the proofreading. Here the proofreading process, that is this is occurred just after the completion of the translation process. Okay, so here I explain that after translation process how the proofreading is occurring. Proofreading means that is the incorrect any bond or incorrect any codon or incorrect any amino acid codon binding, that is binding substance or complex that is released from the chain. Okay, this is called the proofreading. Okay, so I have already explained the translation process, so you just check out in my previous videos. Okay, so here the proofreading is two types. One is the chemical proofreading and another is the kinetic proofreading. Okay, so in case of chemical proofreading, this is used to improve the accuracy of amino acid attachment to tRNA. Okay, so this is what? This is used for the accuracy of amino acid attachment to the tRNA. And this is carried out by the amino acyl tRNA synthetases. This is the enzyme. Okay, amino acyl tRNA synthetase. This is the enzyme that help in this process. Okay, that recognize first the incorrect amino acid. Okay, what is the function of this tRNA synthetase? That recognize the incorrect amino acid, and this is called obviously non-cognate amino acid. Clear? And the correct amino acid, this is called the cognate. So incorrect amino acid from where? Which is already attached to the tRNA molecule, and after that it removed by the process of hydrolysis. Okay, and generally most of the synthetase that binds to the incorrect amino acid, the process is in that way occur, that binds with the incorrect amino acid and form the incorrect amino acyl adenylate. Then binding of cognate tRNA is required for proofreading. Okay, so what is happening here? The incorrect amino acid, this okay, one thing is that most of this synthetase binds with the incorrect amino acid. Okay, synthetase bind with the incorrect amino acid for the recognition, and then proofreading occurs with the cognate tRNAs. That means these are the correct tRNAs. Okay. Next, after binding of the cognate tRNA, when the cognate tRNA they are bind the incorrect amino acyl adenylate, that may be hydrolyzed and released. Then what is happening here? Again I repeat this line, that first of all the non-cognate or the incorrect amino acid, they are bound with the synthetases. Okay, if any sound is coming from the outside, please adjust. Okay, because this is my area is just side of the road. So what happening, that incorrect amino acid they are all bound with the synthetase, and then the binding of the cognate tRNA is occurred, and when the cognate tRNA they are bind, then the incorrect one that are hydrolyzed and released. Okay, and some synthetases, these are unable to distinguish. Obviously there are many synthetases are there, they actually cannot distinguish the cognate and the non-cognate tRNA. Okay, so here the synthetase, these have the two sides. One is the activation site and another is the editing sites. Okay, so activation sites, this is called the synthetic site, and the editing sites, this is called the hydrolytic sites. So what is happening here? Synthetic means activation occurring in that site, and the editing sites means, that means hydrolysis process. By the process of hydrolysis, the non-cognate the tRNA molecules that totally released from the chain, and only the cognate tRNA molecules they remain there. Clear? The second process is the kinetic proofreading. This is generally used to improve the codon-anticodon pairing. This is improve the accuracy of amino acid attachment with the tRNA, and this is used to improve the codon-anticodon pairing. Once the tRNA molecule that have acquired amino acid then form the complex with the elongation factor. So what's happening in the first case? They form, that means the amino acid, they form a complex with the elongation factor, and this complex pair with the appropriate codon in the mRNA. So this amino acid and elongation factor make a complex that pair with the codon of the mRNA. Okay, and this elongation factor, it allows the correct codon-anticodon pairing to occur but prevents the amino acid from being incorporated into growing polypeptide chain. So what is saying here? This elongation factor has important role. What is the role? It allows only the correct codon-anticodon pairing. It helps in pairing of the correct codon and anticodon. And to occur in that way, by pairing of correct anticodon and codon, it creates or it prevents the amino acid from incorporated into the growing polypeptide chain. Okay, it just prevents actual pairing. By the process of pairing, it prevents the binding with the polypeptide chain or entrance or entering into the polypeptide chain, and the initial codon that is recognized first, and this recognition triggers the elongation factor to hydrolyze the GTP that already bound in the place. So the factor dissociates from the ribosome without tRNA allowing the protein synthesis to proceed. This is another important thing. What is saying that when the initial codon recognition is completed, this triggers the elongation factor what? To hydrolyze the GTP. Okay? To hydrolyze the GTP, and the factor dissociates from ribosome without tRNA allowing the protein synthesis to proceed. That means until the dissociation of the factor not occurring, then the translation process or the protein synthesis process is not continuing. Okay, so if the actual initial codon recognition occurs, then the GTP hydrolysis occur and the factor dissociates from the ribosome. Okay, this GTP hydrolysis occur where the place is that the ribosome, where the factor dissociates from the ribosome without tRNA allowing protein synthesis to proceed. This hydrolysis of GTP, again this I am just repeating this line, that the hydrolysis of GTP occur, and when the hydrolysis of GTP occur, then protein synthesis will continue. Okay, thereby elongation factor introduces a short delay between codon and anticodon pairing and polypeptide chain elongation. So in that way it maybe create a delay. What delay? It create a delay in codon anticodon pairing and also the polypeptide chain elongation, and which provides opportunity for bound tRNA to exist from ribosome. That's why the bound tRNA, they get more time to bind with the ribosome. And incorrect tRNA, that molecule form the smaller number of codon-anticodon hydrogen bonds and is more likely to dissociate during this period. So here another important thing is that which are the incorrect tRNA that form the anticodon-codon pairing also, but the number of anticodon codon pairing, these are very smaller. So these can easily dissociate from this period. Okay. So So this is the kinetic proofreading, where the codon and anticodon pairing process is very important, and by codon and anticodon pairing process it creates, elongation factor has the important role, and it creates a time for correction the chain, and until the correction is not occurred, the protein synthesis will not continue. Okay, and here the hydrolysis of the GTP is very important, because hydrolysis of GTP is not possible, then the continuation, this protein synthesis is not proceeding. Okay, and another thing is that no, this one is that too. Okay, from the ribosome. Okay. So this process is the proofreading. One thing we have to check first one. So this the factor dissociates here from the ribosome. Okay, any factor that dissociates from the ribosome. So this is the proofreading, and this is explained very briefly. You just read and from here obviously, and you can check another, you can consult another book that is related to the genetics, so you can properly understand what is this proofreading process. Here I explain very briefly, so you just first this can understand from here and then consult any genetic book. Thank you.